Hello and welcome to this lecture on osmoregulation. This is chapter 50 in Raven et al.'s Biology, 10th edition, which is entitled Osmotic Regulation and the Urinary System. This chapter deals very heavily with how do organisms, and really it's very focused on vertebrates, maintain salt levels and water levels within their body, obviously as this animal no longer needs to do. Again, this is chapter 50 in the book. One thing I would like you to be able to understand is what kidneys actually are. There are also a number of other examples of things that are not kidneys but function in very similar ways to the kidneys in the invertebrates. Those are mentioned on pages 1041 and 1042 in your book. Those are also good to be familiar with. I'm very interested in making sure that you understand how the human kidney works, and so we'll spend more time on the human kidney, but understand there are a variety of different organ systems in different animals. One thing I would like to remind us about that the book does not do is why water is important to animals at any point in their life. So water is a really critical component of life. In fact, it's one of the molecules that we think that life must have to exist, and we think that's probably true anywhere in the universe. And the reason for that is given in chapters 2.4 and 2.5. So if it doesn't immediately come to mind why that would be the case, go ahead and look up chapters 2.4 and 2.5 in the book and read those. But briefly, some of the pieces that make water really special. Water is a polar molecule. Because it is polar, it can form short-term bonds, but it actually forms particularly strong short-term bonds called hydrogen bonds. It has a very high specific heat. It is actually interesting because the solid is less dense than the liquid, so it's inverted. Usually solids are more dense than their liquid counterparts. And water is very interesting because it can dissociate and it can form acids and bases, which changes the solution compound around it. And it can also easily ionize various chemicals that are placed into it. So it's a really useful medium to be surrounded by. It comes with a number of costs and association and issues related to that, but it is certainly one thing that we think life is very dependent on. Remember too that all life, as far as we can tell, initially evolved in water, and they have modified every system that they have evolved with to use these watery environments to live in. So every single living organism, when it is actually going through the process of living, there are organisms that can go into a rest period, but they don't grow, divide, or make more copies of themselves. When they are living, they require a fair amount of water, which they fill their cells with. So the machinery inside of the cell is also very dependent on water. And without water, that machinery simply can't operate. So organisms that do have these long periods of rest are often associated with being able to shut these machine systems down, and they actually have very low or almost non-existent metabolisms because nothing is operating at this time. And if you forget what you're looking at here, this is a picture of those stromatolites from Shark Bay in Australia. These are probably some of the earliest, most complex ecosystems that formed on Earth. They are groups of bacteria that are growing, largely gone from the surface of the Earth. There's just a few places that still have them because things like animals can come along and graze on them and eat them. But at one point in time, this was the biggest and best thing that ever was on the Earth. Some things that you should be completely familiar with before we get too far into this chapter. You should really familiarize yourself with the word osmolarity. Okay, I want you to really understand that. The rest of the chapter will not make sense if you don't read about that. You need to be very comfortable with the words hypertonic, isotonic, and hypotonic. All right, remember these are in relation to something. So keep those in mind as we move forward. And if you don't have a good grasp, if you cannot say it out loud exactly what these terms mean, stop and go look them up. One of the most important components of osmolarity in cells is sodium chloride. So osmolarity is the number of osmotically active molecules in the solute, either inside or outside the cell, right? So this is just in a watery environment. Cells, as you know now and are very familiar with, use a lot of sodium and potassium. There's a fair amount of chlorine, there's calcium floating around, but one of the big ones is the sodium and the chloride, okay? Remember that water diffuses to where ions are more abundant. So the more ions you have in a solution outside the cell, the more the water will tend to diffuse in that direction. The more that there are salts concentrated inside the cell, the more the water will diffuse in that direction. And it is by diffusion that the body is going to control how much water it has in it to a degree. Here I've already used the word isotonic, so again, make sure you understand what this means. What if you're actually isotonic to the solution that's around you, right? So what if you maintain the same level of salts as the solution that's around you? Do you need things like kidneys? Actually, many organisms do this. In fact, most of the what we call invertebrates do this, although it's not universal. There are probably limits to how far you can go, and this image on the right is a really good example of that. And the other thing I want to point out is that things that we call osmoconformers, right, things that modify or don't modify their salinity concentration, are an idea. They're not an exact thing. So if you look at the polychaete worm, which shows a really strong osmoconformer relationship once the salinity of the water gets high, you'll notice that at very low salinities, it actually does regulate its osmotic levels to some degree. Conversely, crabs, or at least this crab, because it varies by different crabs, has a really strong control of its osmotic regulation, internal regulation, up to a point, at which point it adopts a complete osmoconformer. The animals at the bottom there, the eel and the salmon, are most interesting to us in part because they're most closely related to us, and this is in fact what we see in our own bodies. By and large, vertebrates follow a very strong osmoregulation. Even if the solution of water around them is very different from them, they are very strict in the composition of salts within their body. One thing that you can't really do is be an osmoconformer in fresh water. Think about why that is. And just to give you an example, because we're here in the Chesapeake Bay and I wanted to show you some organisms that are around here, notice that the blue crab, which is very common in freshwater and saline environments, is a really good osmoregulator up to most concentrations. At very extreme values, it either becomes an osmoconformer, or in the case of when the osmotic pressure outside begins to become almost fresh, then it has a hard time maintaining salts within its body. But by and large, it's a very good osmoregulator, which shouldn't surprise you since it crosses huge ranges of salinity in its lifetime. Compare that to a mussel, which are usually found along the shoreline and mostly marine environments, then you can see that that's a really strict osmoconformer across the range that it's generally found in. All right, so let's turn to vertebrates briefly then, because I mentioned that they're really strong osmoregulators, and let's think about them in different environments. So in freshwater, one of the issues that you're going to have is you're going to have a lot of salts inside your body relative to the outside. So water is constantly going to be diffusing into your body. Some places that it's going to diffuse in are going to be the skin, but the other really big one, of course, is going to be the gills, right? There's huge amount of surface area. You want the membrane to be relatively thin so that you can extract oxygen, and as a result, that's going to come with the consequence that water is going to be able to move in very, very quickly. So in the picture on the left, where you look at what's called a freshwater teleos, this is true of freshwater fishes in general, then you can see that the blue lines show you that the water is going in. The fish is also, whenever able to, it's pumping salts in from the outside. So it actually has salt pumps that are located in the gills, that if they encounter salts in the environment, they pump it into the fish's body, which makes sense because the fish is always losing salt out of its body, whether it be in urine, or whether it be out through the skin, or whether it be out through the gills, right? So the salt is very, very easy to lose in fresh water. Also notice that the freshwater fish produces dilute urine. So it rarely drinks and it produces very, very dilute urine because there's a lot of water it has to get rid of. Now look at the marine fish. In this case, it has an opposite problem. Just as if you were out in the salt water and you tasted salt water, you would say, this is very salty. And if you drank it, it would dry you out. They also have that issue. They tend to be less salty than the seawater. So the problem becomes that it's very easy for them to gain salts and it's very easy for them to lose water, in this case, not gain water. So they have to drink a lot of seawater, and they have to actively pump salts back out of their body. So they're constantly pumping salts out. In addition to that, they're going to need to produce relatively concentrated urine to make sure that there isn't a loss of water. I'll just point out that this picture on the right shows concentrated urine coming out by the tail. That's not where the anus is located. The anus is actually located almost at the middle of this fish underneath. So this person put the concentrated urine way far back, probably to make it look like a teleost in the freshwater one, which also has urine coming out of what is the tail. Again, the anus is located about halfway along the body. So this is going to be true of the fishes. Your book mentions something about sharks. Sharks use quite a different system for this. They use urea in their body to change the osmolarity of their body. It is good for you to understand why that would change, but I don't expect you to know the differences between, say, sharks and fishes in general. One of the other major components that's going to be excreted by the animal is going to be nitrogen compounds. In the vast majority of fish, that's going to be in the form of ammonia, and you should know the structure and the name. In mammals and in some other organisms, it'll be urea, okay, and that is that compound that you found in the urine. It is not necessarily that yellow compound, but it is present in your urine. If you take urine and dry it out, it will form crystals. Those are urea crystals. The other thing that animals can produce, and this is more common in animals that live in very dry environments, is uric acid. It is very easy to secrete ammonia. It takes energy to make urea, and it takes a lot of energy to convert urea to uric acid. So the actual production of things like uric acid takes time and energy, and so it has to be beneficial to the organism for that to occur. All of these compounds, the production of nitrogen waste, are produced by the breakdown of amino acids, which is very common in all organisms. So you should understand why nitrogen is being produced in these bodies. One thing that is going to be important here is no matter how we cut this up, animals are going to have to excrete stuff, and they're going to have to excrete a lot of stuff, and they're going to have to do it all the time. Things are constantly going to have to be going out. On a side note, this looks delicious. One of the problems, of course, with excretion will be that they have to maintain salts in their body, right, at some very specific limit. Now, even osmoconformers have to worry about the concentration of salts in their body, but they may have to do it by behavioral modifications about where they live. So if, for instance, they live in areas that get more fresh water, they may need to move into more saline areas to survive. So I want you to also appreciate as we go through this chapter that the examples I'm giving you are very vertebrate focused, and of the vertebrates, they're very human focused. Okay, if you're interested in invertebrates, and there's many reasons to be interested in them, you should appreciate that the options there are very diverse and not fully covered in this book. One that I do want you to be familiar with, and again, I want you to understand this not because I'm just interested in you memorizing things, but that you understand how these systems develop from very unspecialized, non-complex systems into very highly specialized and complex systems like we see in humans. And if we just look at humans, you'll get the illusion that these highly complex systems have no predecessors that could ever sort of bridge the gap between something like a sponge, which has nothing like this, and a human. And what I want you to see is that there are lots of these sort of intermediate stages where organisms have some control, but not to the level that we have here. So in the flatworms, which are a good example of a very basal member of the animal lineage, you have these things called flame cells. The flame cells have tons of cilia inside of them. Fluid passively moves from outside, which is the body, into these chambers that the cilia are beating because they're making a current and water is passing along it. And as it moves through, it pulls with it waste material and water, right? And so that moves to the outside. Now, these animals are largely the same salt concentration as the world around them. So the loss of lots of water or what would be dilute urine in a sense, is not a big issue to them, okay? The other thing that these cells will do is that they will actively move material as well into that current and dispose of it. So bits and pieces of other things that have been broken down can be actually dumped into that environment and then expelled out through the animal. Again, this is a very simple system. You can see it branches throughout the animal's body, but it works and is very effective for them. A more complex example evolved in the arthropods, and here specifically in the insects, and these are called Malpighian tubules. Malpighian tubules are really, really important because they help conserve water in insects, which is a really important component of being a terrestrial animal, is the conservation of water. The vast majority of insects are terrestrial. In the Malpighian tubules, they secrete huge amounts of potassium into the tubules. As a result, because the osmolarity of that area changes, water will follow. So it will pass into those tubules to follow these high concentrations of potassium. That is then moved into the intestinal tract, right? And as it moves through the intestinal tract towards the rectum, the potassium is pumped back into the body. And because the potassium pumps back into the body, the water follows. But that is a great way to save water, but it also simultaneously allows you to expel nitrogen, right? So why did we bother putting potassium out to get the water to follow? Well, it was also so we could create a current of water that would bring with it nitrogenous compounds that are now collected into the feces and then can be expelled out of the animal itself, right? So a simple yet complex system that modifies a fairly simple routine of if water moves in this direction, I can use it to carry some stuff with me. All right, so what about humans? Well, humans are very, very, very complex system, okay? They have a very cool place where this is all going to happen, which is the kidney, and we're going to learn about sort of the internal anatomy and workings of the kidney. But one of the cool things that vertebrates are actually going to use is pressure. So in the other ones, you've seen passive diffusion of material after some active pumping. But here we're actually going to use the pressure generated by the heart to push water and materials into a filter and then out. Okay, so that is really quite neat relative to some of the other stuff you've seen for. In a really simple nutshell, this is what the kidney is. A unit of the kidney is called a nephron. The nephron is this sort of beige color. Let me see if I can get my pointer to come out here. This sort of beige color thing here that wraps around, okay? It is surrounded by capillaries and by arteries and veins, and it is going to be very useful to extract materials from the blood. It'll start very simply up here at the glomerulus, which is, I'm trying to find my mouse, is right here. Blood is going to arrive in this area. That is going to lose a lot of water as it does that, the water actually out of the blood. That's going to enter these tubules. The capillaries are still going to retain the blood cells, and they're going to help extract, again, the water back out, but allow things like waste to be removed. Materials that you do want are going to change over time. Sometimes you're going to want to retain salts. Sometimes you're going to want to expel them. If you had been alive a few hundred years earlier, this would not surprise you. There were people that tasted urine as part of a medical practice, and they could easily tell you that urine tastes different at different times of the day. But in the case now that most people are not routinely tasting urine, thank God, that this is less obvious. But your urine changes pretty frequently, and that includes not just the color and the amount, but it includes a number of salts and the material that's actually in it. Everything else that is not recollected will be sent down here. Let's see if I get this. And that will be expelled out as urine. So that heads towards the bladder. And there are many, many, many of these nephrons inside of your kidney. Again, one of the very cool things about the vertebrates is vertebrate kidneys can make isotonic or hypotonic waste, which is cool. But birds and mammals actually can make hypertonic waste. So they can make very concentrated urine relative to their body. Again, they don't always make super hypertonic urine, but they can make it. So if you have not had much to drink for a long time and then you urinate, you will find that your urine is very, very dark and you are making a very, very hypertonic urine. If, on the other hand, you are sitting at home and doing nothing but, let's say, drinking coffee and tea all day, what you will find is you're going to make a relatively isotonic urine or a hypotonic urine, probably because you're losing so much water, right? The amount of water is being removed from your body at very high rates relative to normal. Now, I have chosen not to actually go through this in detail, in part because there is a phenomenal crash course on the kidney already present online. I will link it here, and then I'll put the link also in the description. This is really a well-made part of the video. It covers very closely to the material you'd learn in the book about how the kidney operates, and I do expect you to watch this one. All right, if you've already seen that video, you're back here now. One of the things the video does not stress, which I think is really important to understand, is that the kidneys rely on this countercurrent exchange. And as I mentioned when I introduced countercurrent exchange in the respiratory chapter, countercurrent exchange is very, very useful, and it's used throughout organisms, but it is not just for oxygen, and here is a great example of that. Look here in the loop of Henle as this relatively dilute, what will become urine, starts to travel in, so it's the water and material that is in it. Look at how it's able to pull water out because there's a strong countercurrent exchange. That will highly concentrate the salts in that waste, and then the salts that are wanted back in the body will be extracted, and then the urine will be passed out. What will then become effectively urine will be passed out of this body. So there's a section of it here that's primarily focused on the extraction of water. And then there's a section of that loop that's primarily associated with the removal of salts. And because of the way they're set up with very high concentrations working in opposite against each other, you get strong diffusion the entire length of the loop of Henle instead of reaching some equilibrium point. If it's confusing to you what I'm describing, what I would like you to do is really spend some time on pages 1048 and 1049, where the book goes into real detail about how water and salts are extracted. One of the things I would like to introduce you to now is the idea that there are such things as hormones, which are chemical messengers that can be sent from distant locations in the body to other places in the body to cause changes in those different systems. In the case here, if there are interpretations that the blood pressure is rising too high, aka the osmolarity of the blood is becoming too large, then what can actually happen is this thing called antidiuretic hormone, ADH, is released into the bloodstream, and this will go and change the way that the kidneys act. ADH will change the way in which water is collected in the kidney, and here what it does is it causes aquaporins to be released into the distal convoluted tubules so that more water is retained within the body. So as you become dehydrated, the kidneys do a better job of retaining water within your body. And on your right, what you're looking at is an example of how urine changes. So just to convince you that there are real differences here. And these differences are characteristic of a lot of different things, but they are also good examples of when systems are either failing or there's some problem internally, and is one of the reasons that it can be such a useful diagnostic. The conservation of water within the kidneys, of course, is going to follow a negative feedback loop, as you should expect at this point. Very simply, again, what I just stated in the prior one, osmolarity of the blood will increase. The osmoreceptors within the blood will detect that. They'll send that information to the central nervous system. The central nervous system will produce a thirst cue, right? And simultaneously, it will release ADH. ADH will trigger vesicles in the cells with aquaporins to be sent to the distal convoluted tubules, where they will bind to the surface of these cells and distribute more aquaporins. Aquaporins, because they're pores that water travels in, will allow water to easily move back into the body. So it will do a better job of trapping water as it's trying to escape. All right, that concludes our lecture on this osmotic regulation in the animal bodies. Again, because presumably you've looked at the crash course. I also want to remind you that exam 3 is coming up, and remind you that you need to spend a lot of time on this material because a lot of it relies on a different set of language that you may not intuitively understand. So you're really learning two things simultaneously, the science of how these systems operate and the language of biology. Okay? If you are looking for additional tools to use with your book, remember that there are learning outcome reviews at the beginning of each or at the end of each section, which you should take a look at. There are things usually within chapters called scientific thinking. These are very useful little checks to see if you understood the material within it. And then there's a very nice end of chapter review or chapter review at the end of each chapter, which is effectively a study guide for that chapter summarized in one page. And finally, in the book itself, there are review questions. These review questions are usually very reasonable. They come with a background from very simple to more complex. And so if you really want to see, do I understand the material, go ahead and take a look at these. Again, there are some in here that go up to very complex levels, and if you're easily able to answer those, you'll have no problem when you get to the test.